Good afternoon, everyone. I think we'll be starting on time and the rest will join us along the way. Thank you for joining us today in the May seminar. And today we'll be hearing from the FIPS project and it will be Dave, Priest and Vincent Wandera presenting to us about their research data life cycle. They'll be sharing their experiences, what has worked for them. So I'll be handing over to Dave. This is a recorded seminar and the recordings will be available as well as the presentations in the Dropbox folder that I will share at the end of the seminar. Over to you, Dave.
So yeah, Vincent and I are talking on the research data lifecycle. And when Schiffer asked us to present, she shared this, this wonderful cycle here, talking about all the stages in research, which felt to us fairly daunting and intimidating. So we had to think about which parts of it does FIPS have something to contribute or share with the wider CCRP network. And it felt as if maybe the best areas we could share on our plan and design, collect and capture, analyze and share. And I think our partners from FRN NGO are going to also give some ideas on other parts as well. We sometimes internally simplify the research data lifecycle into just plan, do, review. It's a very simple thing to just remember in our head. We can say to ourselves, plan, do, review several times a day. So before we joined CCRP, FIPS was an organization that was really on the front lines of sharing information with as many farmers as possible. And we were doing experiments with farmers, but we weren't collecting any data. Our values really built on they were really sisters to the CCRP values of understanding farmer constraints, building on farmer practice, and combining global and local knowledge. We also believed very strongly in learning by doing, adapting, changing, in which as many farmers as possible should be given the chance to try on their own land using small packs of seed. Largely, we work with simple things like seed. And this would allow hundreds of farmers within a few villages nearby each other to all experiment on their own land. And they would all have their own data points. This meant the scale of what we were doing meant we had to keep it simple. And we'd go back and we'd review what works, what's working, and if something was working, we'd do more of it with more farmers. If it wasn't working, we'd stop. That was our previous research data lifecycle. Data really looked like going and visiting farmers or taking pictures, where you would compare what they were doing before and what they were doing afterwards, either as a result of adapting or trying new options. So changes like this, as you can see on the screen, to us were data points, and they were saying, this is working. Farmers were telling us it was working. They were going on to try and adapt what they were doing on larger parts of their farms. And that was our data. And then Rebecca came to visit. This is a picture of Rebecca having a go doing deep tillage using a modified ox plow in eastern Kenya. And she looked at what we were doing and she saw, wow, in a single village, I'm seeing more than 20 different types of experiments with over 200 farmers, and FIPS are working in 500 villages. We're collecting names and information about farmers, 100,000 farmers, and we're doing spot checks with 5,000, but that data is giving us almost no value. And Rebecca was just asking us, how on earth are you learning from this? when you're doing so many things and you're not collecting data. And she asked the other question of how much more would you get if you had more of a research data life cycle? And so we took up the challenge and we decided to have a go at improving by learning with Rick and Rebecca and the whole CCRP team. So within our plan, do, review world, was there even a problem? We had to ask ourselves that. The old FIPS could test simple problems such as an option, such as varieties. We could understand and adapt together with the general farmer, and we could share information with 100,000 farmers. But what we couldn't do is do much with complex problems. We couldn't understand where things were working or not, or for whom, and we didn't have the confidence to share what we were learning outside of FIPS because we had nothing empirical, no actual data to back it up with. We were concerned that research might take too long, it might not value people's time, that we needed to keep things simple. And Rick told us, don't worry, you can do all of that, but still be more reliable at research. The other value that we had was we wanted to say all of our research, we need to say, once we know the answer, what will we do differently with the information? Because when we do research, we want to make sure that we're learning things that can help us share and adapt with more farmers. So we developed our own research data life cycle. The first stage was planning and asking questions, and we developed this framework of starting at the bottom, what is likely to work? Often we see research programs where they spend a lot of money doing beautiful research on something that you could probably guess before it started would never work for the type of farmers that we're working with. Then we move up the cycle into what is working by doing small N studies, where is it working and how much by doing medium N, and for whom are they working by doing much larger N now with thousands of farmers to see which sociological types of farmers are adopting. And at each stage, together with farmers, we would adapt and improve the options. Some simple tips. First of all, understanding farmers, valuing their time and not experimenting with things which are unlikely to work. When we think about what's likely to work, we think about, is it cost effective? Does a farmer have the cash or the labor to be able to do it easily? Does it lead to improved gross margins for those farmers? Does it technically work? Is there a way that they can get the things that they need? And also considering other things like poverty, gender roles in relation to particular options. We also found that we had to know our end in mind. What are we trying to achieve? After doing this research, what would we do differently? And we ask that question more than what is interesting. Sometimes we ask what's interesting, but often we just start with what would we do differently if we know this? Examples of what might be unlikely to work. This is a farmer carrying a bag of fertilizer on his bicycle to his shamba. Now, if we think about liming, you need 800 kilos an acre, according to conventional recommendations, which would be 16 bags. This picture here is 13 bags. Imagine carrying 13 bags on the back of that bicycle. We don't even need to think about it. It's not going to work, or it's unlikely to work, let me say, for most farmers. Another example is people going around saying spray chemicals to control fall armyworm. When we know most farmers that we talk with don't own knapsacks, they don't have a culture of spraying, they don't know how to spray safely, they don't have PPE, and they don't have cash flow to buy the chemicals. Even if we thought it was a good thing, it would still be very unlikely to work for many farmers. Another example is fixed time artificial insemination in farmers where the cows look like this. You can see the cow is totally emaciated, it's skinny, it's not going to get pregnant. So doing experiments around improving AI for farmers with cows is going to be a challenge. And let me say, we have made all these mistakes ourselves. So we're not being arrogant, but we try and now learn and be better by asking what's likely to work. We also think about what's actionable versus interesting. In the example of fall armyworm, we became aware of the soil experiment, the soil method, which I think we've shared with most of you before, of putting soil into the whorl of a maize plant, and then that controls the fall armyworm and leads to less growth. So we ask the question, what will we do if we know it? Is it likely to work? And for us, that's the important question. And if farmers know this, and it's cost effective for them in terms of the amount of energy required to do it, then it's likely that we can share with many farmers and they will adapt and improve it and go ahead and do it. Questions which we think are valid questions, but we might not ask, might be, what is the mechanism by which soil kills fall armyworm? Those are very valid questions, but they're questions which we in FIPS tend not to ask. So I've talked a little bit about plan. Now I'm going to talk a little bit about collecting data. So first of all, we have to ask the question, what data do we want? And when we think about that, the number one criteria is valuing people's time. I think we've all been in situations where we have a 2-hour survey or a 1.5-hour survey, and by the time you're halfway through, the farmer's looking out the window and bored, the enumerator is bored, half the questions are very difficult to answer, so take a long time. And that collecting that type of data is unlikely to be reliable data, and it's unlikely to be efficient, and it's not valuing people. So in the early days, we used to just try and collect everything that might be useful or interesting. We realized then that we never even analyzed half of it. We never took the time to do much with it, and maybe a quarter of the data was used if we were lucky. So now Rick showed us, start off asking, what do we think we need? Maybe we think we need to know yield data. Then we can ask, how will we use this information and what will it tell us? Maybe we'll make a graph comparing yield using the option versus the control, and it will tell us, is the option better than the conventional practice? So what do we really need to know? Actually, what we need to know is it better than conventional practice? Is it, and maybe not just a little bit better, but is it a lot better? And is it consistently better? So to answer those questions, we may not need yield. We may just be able to work with Likert scales, because ultimately what we want is for the farmer to see whether or not this is worth doing and make decisions about how to adapt it if it is worth doing, or not do it if it's not worth doing. What will we do if we know this as well? Maybe we will share with farmers. We'll look at value cost ratios, but for that we'll need yield. So there's lots of questions that we go around asking ourselves before we go out and collect data. Another important question that Rick taught us was, what data do we have anyway? This might be things about gender and age, which we have to collect for our projects when we're reporting to, for example, USAID. So we have information that we've collected from 100,000 farmers about their gender and age. So how can we use that data? Then also another question would be, what is easy and reliable to collect? We can easily get GPS points, soil pH, using Steve Vanek's soil pH meter. We can look at texture, soil texture. We can ask simple context questions like, do you own a knapsack? What type of cow do you have? And use them as proxies for wealth. It's much more easy than asking complicated questions like, how much money do you have? How much money are you earning? We can, that takes a long time and you never even believe the answers. Another question, what could this information tell us? In this case, FIPS has been using these types of data to understand who is benefiting from the options that farmers are adapting. We still ask the question, what else is interesting? And in some cases, we will ask something that is less actionable, but we just do for pure interest, such as how much phosphorus is released by the microdosed lime in a lime study. Then when it comes to collecting data, again, we need to think about valuing people's time, as I'd mentioned before. And that's the time of the farmer, the time of the enumerator, the time of the people doing the data crunching, everybody. And we ask these four simple questions. How many replicates? Who do we collect data with or from? Who should do the collecting? And how do we collect? And within that, we need to consider different things like, well, how many replicates might we need? What is the type of data that we might be collecting? Is it numerical? Is it yield? Is it categorical? Is it a comparison? Could be many types of data. We also need to consider the sophistication of the people who are either being collected from or who are doing the collecting. And we also have to consider who benefits from that data. Within how many replicates, we might want to think about how easy is it to collect. You can collect many more replicates if the questions are simple. How many farmers can you visit in a day is a critical question that we ask. Maybe someone can visit 10 or 15 farmers in a day. And the other thing is, what's our failure rate? If we lay 300 trials, how many of them do we expect to fail because of flooding or poor management or the farmer's not available or some other problem? Who to collect the data from? That could be the farmer, could be the FRN farmer. In some cases, when anything about money and prices, we find it easier to collect data from the market centers nearby. What's the prevailing price of this? Much easier than asking a farmer how much they sold it for. And you only need to collect it once because it's probably valuable for a range of areas. Who can collect? That could be your FRN farmer or a village-based advisor, could be field staff, it could be interns, depending again on all of these things about the type of data, the sophistication, and who's benefiting. We tend to say if a VBA or an FRN farmer is not going to benefit from the data, we should not ask them to collect it. So there are certain types of data which really are for our benefit or a funder's benefit. Then we say we're not going to burden the farmers involved in collecting that. They only collect data that they themselves will benefit from. How to collect? Many things. Paper, advanced forms of paper, which we call logbooks, where we pre-print hardback logbooks, and the farmer then can keep the copy of all their data on their farm. So, and we just take photographs or photocopies of it, rather than taking away the paper. ODK is something that we've learned through CCRP and the Research Methods Support Group. Now we use that routinely within all our work. And we've experimented with collecting data using USSD or SMS shortcode, where we send out messages to farmers and ask them to do the press 1 if you think this, press 5 if you think that. Let me give you the simple answer. We got very, very low response rates. It's anywhere between 2% and 11% from that. So we have stopped, or we're stopping using this as a method of data collection. So that's about data collection. The next topic that we wanted to discuss was analyzing and interpreting data. And the first part of that we've called the art of drawing. This is the whiteboard in our office with lots of pictures and drawings. And I think this is a skill which I learned from, again, some of our mentors, Rick and Rebecca, is that whenever, and I'm sure many of us on this presentation have had a similar experience, is that when you're sitting discussing a problem or an issue, and then you, with Rick or Rebecca, and you see them just drawing 2 axes and splitting data up and saying, wouldn't it be interesting? And they start drawing pens and pencils across the board. And what I've learned is by drawing things out, we become a lot better at thinking through what is the analysis that we want to do. And it's much easier to draw things out several times over and play around with them and do various iterations of them than it is to actually make graphs. It's much easier to start with the drawing and then organize your data to be able to create the graph that you want. Things that we think about while we're drawing is, well, what were our hypotheses in the first place? What might this, or otherwise, what might these data tell us? Another thing to think about is, when do we draw? I would love to say that in the planning, all this drawing happens and we draw out and we think about our hypotheses and all the ways we might present the data. And occasionally that does happen, but very often it happens at the point when we've got data in and we're thinking, what's the best, clearest way to make sense of the data? But it's definitely better to start early, but keep drawing throughout. And the other thing to think about is, what are we trying to communicate? Is it that we're trying to understand something ourselves, or are we trying to communicate something with someone else? And what is it? And who are they? Because what we draw and what we present will be different depending on who our audience is. So if I give an example, I think many of you are aware of our work on using soil to control fall armyworm, and that we're comparing it with chemicals. So we had done a survey of around 5,000 farmers, I think, to understand who is using soil. After learning and being trained, who is using soil and who is using chemicals. So we drew out here percentage of farmers on the y-axis, and then we have we're expecting more farmers to be using soil and chemicals and fewer farmers to be using other methods. Then when we thought about it a bit more, our hypothesis was that people with a local cow, people with no cow are poor, and they don't have money for chemicals. So this NC, they're going to be using soil the most. People with a local cow have a bit of money, but not a lot of money, and they'll still be using soil. And then people with a cross cow or an improved cow, they probably have plenty of money. They're probably comfortable using chemicals. They probably have a knapsack because they control ticks on their on the cows. So they're probably going to be using chemicals, so very few of them will use soil. And this was what we thought would be a proxy for wealth. Similarly, we thought farmers who don't own a knapsack, that's minus K, would be using soil in high amounts because they don't have the option of chemical. And farmers who own knapsacks would probably not use soil because they can easily just go out and spray. Similarly, with regard to soil, we had the question of, we thought people with household size, people with many farmers in the household would be using soil. The higher proportion of farmers with big households would be using soil because there's plenty of labor. And people who have small numbers of people in the household who don't have much labor available, they would find using soil difficult, so there'd be few. Similarly, we thought people with small plots would find it easy to use soil on a big area, not to use soil on their whole area because they've only got a small plot. And people with big plots of land, they won't have enough labor to put soil on all the maize all over the plot because it's labor intensive, so few would be. Then we thought, we can put chemical on the same plot as this. Instead of making it percentage soil, we could just make it percentage farmers. And this is the graphs redrawn now, where blue is people using soil and red is people using chemical. Communicates a similar thing. Another example of the same thing is that we thought about using let me go back. We were saying, how do these two things interact? Number of people in the household, we didn't know what is many and what is few, and what is a small plot and what's a big plot. We thought there would be a line somewhere. So following the Rebecca approach, we drew it on two axes, land size out here, family size up there. And then people with big land and small families would use chemical, and people with small land and big families would use soil. And probably there'd be a line somewhere in between. So if we plotted out all the data points, we'd probably find all the soil people up here and all the chemical people down here. And we're interested to know whether that was the case. The next section of my presentation is using care with color. Here's a graph which came up as using all the default colors on Microsoft Excel. And it's really difficult to understand what this means. You've got the legend down here. You've got all these colors. So gray means, oh, dry soil. Okay, I'm struggling to understand that. If we're a bit more careful, first of all, never use the default. Putting the legend at the bottom is complicated for a graph like this, and it's not intuitive. This is much more intuitive, where the soils are brown. So people using soil paste and dry soil are both shades of brown. Chemical is like a danger, so we made it red. And ash is gray, so we made it gray. And then this is a thing, I don't even have to refer to the legend because I can easily remember these colors because they're intuitive. And the other tip I've done here is I've put the legend up the right, so I can see soil down at the bottom, chemical in the middle, ash at the top. And I can even simplify it. The other thing I've done is I've shaded the colors. So 2 types of soil with 2 different shades of the same color. And even in another version, I just realized I don't care which version of soil they're using. So I'm just putting all the soil types in the same gram. And the next thing is just be very careful with color blindness. 10% of Caucasian men and some Africans, but much fewer, can't tell the difference between red and green. So they would see traffic lights like this. And instead of seeing a red and green plot, they might just see 2 shades of gray, which is much harder to understand what it is. And if you have more bars in your graph, it's even more complicated. Again, care with color. This is the way that we presented Likert scales, where we had a 5-point scale going from blues are a lot better or slightly better. Green is about the same. Green is a neutral color. And yellow and orange is shades of red, again, bad. So they're on the yellow and red. And this is using shades, 2 shades of yellow-orange for worse, and 2 shades of blue for better, and green for neutral. And once you've understood this and explained and seen that blue means that farmers have a thicker stem, you can, I'm just sort of, oh yeah, I've explained the shades, I've explained the legend is up the side, I've explained color blindness, so we're avoiding using reds and greens in this. And the other thing is, if you do another graph, this is now a different characteristic. This is about maize cob size, but we're using the same coloring. And by using the same color coding and being consistent, it's much easier for someone to jump and understand very quickly what the next graph is telling them, that most of these people, things are better. Things are better for most of these people because they're blue. But never forget the value of illustrating your data with a picture. As much as all these graphs in the world tell us something, this picture tells us more. Using soil gives us bigger cobs than not using soil. So we've talked about the care of color. Now I'm just going to finish up with talking about some of the things we think about with sharing data. We titled this section, Caring Means Sharing. If we care about our research, if we care about our farmers, if we find interesting results, we have a responsibility to share it with the wider world. But when we're sharing, and in FIPS that means sharing with as many farmers as possible so they can improve their lives. And that they can continue adapting what they have learned as well and improving further. So when we're thinking about sharing, we have to first think about 2 questions. Who is it for? And what might they do with it? We might be thinking about sharing information and data with FRN farmers or normal farmers. We might be thinking about sharing with project managers.